Today we got Dr. Peter Yoder joining us, currently a technical services manager for Perdue AgriBusiness Animal Nutrition. Peter grew up on a small dairy farm in central PA. He got his BS from the Pennsylvania State University in Animal Sciences back in 2010. I was lucky to spend my time in my master's with him in animal sciences at OSU and finished in 2012. And then after a couple of years in industry, went back for his PhD in dairy science from Virginia Tech in 2018. So Peter's graduate research has focused on dietary nutrient variation and amino acid metabolism. Peter enjoys spending time on his family's farm, international travel, and learning about other cultures as well as volunteer work. So at this point, Peter, I'm going to turn it over to you. And we were talking before we got started here that there's basically 3 different sections to your talks. So folks, if you type questions in the live chat as we roll, then every so often Peter will stop and we'll clear through those questions. With that, it's on to you.
Good afternoon. I appreciate the introduction, Dr. Winter, and I appreciate the opportunity to present here today. Humbled by the opportunity and have several different subtopics I'd like to go through, but the general theme of the talk is related to formulating diets for amino acids with changing milk prices. Should we be changing how we formulate diets? The first slide here, this is just an overview. This is Federal Milk Marketing Order milk protein prices, January 2009 to April 2020. We just got April prices last week. I think it was $2.48. What you can see though, over the last, say 10, 11 years has been a trend downwards, particularly this 2015 to 2018, milk protein prices were depressed. We have an increase here, and then we've seen a decrease, particularly in the last several months here. Should this affect how we formulate diets is one of the areas I'm going to come back to in a bit. This is looking at a smaller section of that data range. I'm looking at January 2019 to September 2020. The last 6 months here, this is actually supposed to be May. Axis didn't line up right, but this is May, June, July, August, September, October. This is a futures price for milk protein based off the futures for butter, cheese, powder, the 4 classes of dairy commodities. So we're projecting an increase, a pretty good increase, and we could be up to $3 here by September. What I find interesting is if you look back for, say, early 2019 versus where we are today, it's roughly the same, if not higher. So we are seeing somewhat of an increase here in milk protein prices, despite milk prices being low. And that's because of the fat portion in cheese is subtracted out. So once you know the butter price, you subtract that off, and then you get the protein price. With butter fat crashing, exports crashing, that's helped elevate milk protein prices, as you can see right here. So what's the value of a 0.1 percentage point of milk protein? Just for rough calculations, I assumed 80 pounds of milk. And I just want to look over historic here. This is the value. You can see here in 2015, we're about 18 cents. 2020 year to date, we're close to 23 cents. Futures market is predicting 21. I don't know if you all realize this, and I was sort of a bit surprised when I put this all together. 2020 may be, based on our futures market, now that's expectations, the best year for milk protein since 2014. You can see here in 2020, if you take 6-month futures, where we're at year to date, we may end up around 21, 22 cents for this amount of milk protein on 80 pounds of milk. So definitely, I think for some farmers and some nutritionists, this might be a surprise seeing this change here. You open up your milk check for April or even May, milk protein is going to be the majority of the paycheck, and butterfat is going to be the minority. So here's just a rough diet example. I'm going to come back to some examples as we go throughout to try to make this somewhat applied. This is just, as say, a typical diet in Upper Midwest. I didn't try to do anything special, but I just want to put it out here. Say they're formulating for 90 pounds of milk, 16.4 crude protein, balancing amino acids according to the CNCPS model. This is NDS software, by the way. This is the cost per calorie day at 56 pounds of dry matter. If you take the blood meal in this diet, the rumen protected amino acids, and heat treated soybean meal, that's $1.40 a cal a day. So given the milk prices, we're expecting to see milk protein starting to dip a little bit, but then going back up. Should we be changing how much we're feeding of these? Should we be formulating to what the model suggests is the requirements right here? And I'd like to get into this in more detail throughout the talk. Is, do we need to be exactly at this number, these requirements, or could we be formulating for less? Just for review, CNCPS, they have several ways of looking at amino acids, either by factorial and the requirement, which is shown right here. So I'd have 108% of requirement for methionine, or looking at methionine versus energy, mcal, and being 1.16, where the optimal would be 1.17. So the other thing, we have changing milk protein prices. As I said earlier, 2020 may shape up to be above average milk protein prices compared to the last 5 years. We also have changing nutrient prices, and we have changing feed ingredient prices. This is blood meal over here on the left. You can see this is from beginning of 2019 to today, or last week. As you can see, there's been a steady incline, particularly here since November. Blood meal is widely used as an amino acid supplement. The price is essentially nearly doubled, 40% increase, or nearly doubled in price since November. Does that mean I should take it out of my diet? Does that mean I should feed less? How should I change how I utilize this ingredient? The other thing, and a better way to look at it, is using sesame, where we look at the cost of nutrients and what feeds are a bargain, priced appropriately or expensive. As you can see, as blood meal is going up, when I look at the general aggregate of metabolizable protein using ingredients such as expeller soy, the overall average is going down. So this is from January to March here. We see a trend down, did see a slight increase here, but relatively flat. I would say with where soybean prices are and some of your other protein ingredients, it should stay pretty stable here. So again, there's these changes in nutrient prices and there's changes in ingredients. And when we formulate diets, we probably need to make adjustments on how these things change. This is a very small study I did several years ago, and this is just a concept, and I'll get into this concept here in a couple more slides. I looked over a 2-year period, 2014 to 2015, and looked at the range of milk prices, the range of feed ingredient prices by month. Then I took the highest feed ingredient month and the lowest milk protein price month versus low feed, high milk protein, and looked at what is the optimal diet formulation. I used a response-driven equation, which I'll point out in a bit. But what you can see is, as I went from high feed to low feed, the amount of metabolizable protein increased. This is going from 2,400 to 2,700. And this is for a 45 kilogram cow. So I see this increased metabolizable protein. As milk prices change, I also see I could feed more. So my point with all this is, as we see changes in nutrient costs or protein or milk protein prices, we may want to change how we're formulating to diet. This marginal MP efficiency, which we'll get into that a little bit more, you can see that decline. So when feed is expensive and milk protein is low priced, I have the highest efficiency, over 20%. As feed gets cheaper and milk protein's higher, I feed a lot of protein and I end up with a lower marginal efficiency. So a little bit about this diet optimization and follow up on that. Most of us will look at optimization or least cost formulation in this framework right here, where we're going to enter in ingredient costs, say such for soybean meal or blood meal, etc. And we're going to set requirements. So we're going to say we need this many megacalories of net energy, or this amount of MP in grams, or this much methionine in grams. And then we use an algorithm, a least cost simplex algorithm, and we come up with the least cost to meet that nutrient requirement. I think some of us will assume this to be maximum income over feed costs, and I would really challenge that, because how do we know these nutrient requirements are distinct in a single number? If we feed more MP or we feed less MP, what happens? The assumptions of linear programming are proportionality, additivity, and certainty. With proportionality, you assume a constant efficiency. NRC, CNCPS has an efficiency of 67% for metabolizable protein. So that holds if that's true in biology. The problem is we have a diminishing response, and that's not true. You'll see from data I show that you don't see that hold. Additivity is interaction of nutrients. If I feed more starch, or if I feed a certain level of RDP versus a higher level, additivity assumes no interaction of nutrients. That's not going to hold in ruminant nutrition when we're least costing. Then lastly, certainty. We're assuming that we know ingredient specs with certainty, and we're also assuming that we know requirements with certainty. And I would suggest we likely don't, and there's uncertainty surrounding that. So another way to look at this as far as maximizing income over feed costs is, okay, so what if we consider milk component prices? What if we consider a nutrient response? So there's a response surface. So as I feed more protein or less protein, how does that change milk protein output? And that may change depending what I should target based on milk component pricing. Additionally, nutrient costs. This is very similar, or ingredient costs, which is very similar over here. So it's gonna put together ingredients to deliver metabolizable protein or energy at least cost. But here, if MP is expensive, I may feed less MP. In this situation on the left, I got to feed to requirement. If I revert the model to not being requirement, as I'll show in a bit, the marginal response surface is very different than what we see in cows. So again, this is where I think we should be looking at diets, considering these four factors. However, our nutrition models today, I don't think enable us to do that, which we'll get into here in a bit. So a little bit about our nutrition models. And I'm primarily going to be talking about NRC and CNCPS today, CNCPS version 6.55. This is a general comment. I think looking at data out there, predicting metabolizable protein, there's been significant work and research gone into that. And some may argue that it can be predicted with fairly reasonable precision and accuracy. Some may argue not, but you can predict supply of amino acids, supply of the aggregate in MP, and then testing using these computer models. However, both of these models take the absorbed metabolizable protein and multiply it times 0.67 to get the predicted milk protein yield, and then to predict milk yield from that based on concentration. So if I change the amino acid profile, if I have more methionine, this efficiency doesn't change. If I'm overfeeding protein, we know this efficiency would be less. This doesn't change. If I change the energy content in the diet, this doesn't change at all. So this is a fixed efficiency regardless of the profile of that MP, regardless of the interaction with other nutrients, regardless of the stage of lactation. And so this, looking at it on a response surface, may lead to estimates that are very different than what we would observe when we do research. So essentially, when we get to supply and converting it to milk protein in our models, post-absorptive metabolism is essentially considered an inflexible black box. This inflexible black box, I think, makes it a little difficult when we talk about the optimization, as I did on the previous slide. So a little bit on requirements. and just this is more, I guess, food for thought to think about. Do discrete metabolizable protein requirements exist? Is for, say, 100 pounds of milk, is there a lysine requirement? Is there a metabolizable protein requirement, or is it a response surface? And I would suggest, and we'll get into this a little bit, given where we are in efficiency, this is a typical gross nitrogen efficiency we see. Metabolizable efficiency may approach 40–45%, but overall efficiency is low. So we have a lot of excess amino acids, excess protein that's going to be excreted as urea. If we keep lowering the protein, and we see this in research, we see changes in performance. Is that because of nutrient signaling? Is there certain amino acids that stimulate milk protein that change the rate of protein synthesis? And I would argue yes. And so we have this response surface where as I feed more nutrients, something happens. It just doesn't appear. Energy is a little bit more simpler to explain. In this case here, if an animal would consume excess energy, or we as human beings consume excess calories, something happens. When a cow consumes excess MP, I think most of us think it just goes out as urea, milk, urea, nitrogen, urine, or in fecal. But I think there's also a nutrient response surface there as well. And so I would agree with this comment here from this paper that it's likely impossible to derive discrete nutrient requirements. Here, this is a review paper where they looked at a previous study by Dr. Glenn Broderick, and I thought this illustrates this point well. The black line here is observed. This y-axis, I apologize, not going to use, is milk protein yield, grams per day. The orange line here is CNCPS 6.55 predictions, and the blue line is NRC 2001. So what you can see is across these different levels of crude protein, looks like 16.5 is the optimal. We're seeing a small increase here. You see a little bit of a decrease and just sort of flat here. But this 16.5 seems to be optimal. NRC, you have this linear increase. So as I feed more protein, more MP, the model is going to predict more milk protein yield. CNCPS is very close to what was observed here, but you also see that same trend in the data. There was also a reduction in dry matter intake. I think that's part of the reason you're seeing this here. But when you look at just how the models compare to what's being observed, low protein diets, NRC missed by 22 pounds. You get to 15%, it's a little bit tighter. Here at 16.5, NRC is still missing by 7, CNCPS is over by 7 pounds. So that means is to get this level of performance, the CNCPS model, perhaps you need to overshoot by 7 pounds MP milk. When we get out here, you can see large increases as well. So again, pointing this fixed efficiency in the model can mislead us on the effects of marginally changing protein supplementation. This study is a little bit dated, and you can tell that here on the y-axis. High producing herds will be 1,500 grams per day of milk protein. You can see in this data set, the maximum was about 1,050. But what you can see in this data set is that the response surface is changing. So the first derivative here is 0.4. Out here, as I get more MP, it's 0.2. CNCPS is going to estimate 0.67. And then once we get to the requirement, it's going to be flat. The same for NRC as well. So again, just showing that this efficiency is going to vary. So I believe most of you probably thought about this or maybe seen this, but this is maybe somewhat review, but I like this simple slide in that if I have 100 grams less MP or 100 grams MP increase, if I'm using the NRC model or CNCPS in a 3:1 true milk protein, I'm going to predict a 5-pound loss. So I would predict 5 pounds less milk production. Let's say $12 a hundredweight. I'm going to predict 60 cents a cow a day less revenue here. When I overfeed MP, I'm going to say no response because I'm saying above requirement, there's no response. If I take this data set I just showed you, that response is going to be very different. In this case, that'd be minus 1.4, which is again, about 20 to 25% of this response. And I'm also going to predict an increase here. So knowing this may change how we decide whether to pool less blood meal or feed more blood meal or feed more heat treated soy or less, depending on the prices of metabolizable protein. So to conclude part one here, the stress optimal nutrient supplementation for income over feed costs likely varies. We need to consider prices of milk components. So when milk protein is high versus being low, you may want to change how much metabolizable protein or how much of specific amino acids you're feeding. The same goes for cost of nutrients. The marginal response predictions by models are likely inaccurate. So what I have here is this would be for May using $1.98, which this would be a futures price here. And I'm going to say I have the models, which would be NRC or CNCPS. If I'm going to feed 200 grams less MP because I want to save money on feed costs, the model is going to predict I lose 134 grams of milk protein. That marginal revenue, so if I use this $1.98, that would suggest a 58-cent loss. I have a feed savings, both in terms of MP, and I also accounted for the energy cost of this milk protein. So my marginal return would be a loss of 38 cents. All of us looking at this would say this is the wrong decision to make. We should not do that. However, if we look at this estimate here, say the same thing, 200 grams, this model using a 19% marginal efficiency would predict a 38 gram loss. This is based on this more recent paper here by Daniel et al. Marginal MP of 19%, which is sort of where a number of papers have found the marginal efficiency to be when you're feeding close to suggested requirements. So minus 38 grams, this is a change in marginal revenue. If I account for MP cost and the energy cost of this 38 grams, this is basically break even. So if I do want to decrease 200 grams to reduce production, from a marginal return standpoint, is probably going to be the same as feeding, not changing MP. Vice versa, if I look at increasing MP, of course, in these models, NRC, CNCPS, there's not a positive increase, so you'd have added feed costs, so 16 cents in added feed costs. Versus if I look at here, if I want to increase production, basically the same, it's basically break even to minus 1 cent a cow a day. So I would encourage you when you're looking at diets, for example, this diet right here, 97 pounds MP milk, 91 pounds ME milk. Does that really mean excess 6 pounds or 5 pounds of MP milk? I would argue no. I would say you are a little bit on the high side using this model here that I just described. That's the effect if you would reduce this 96, 97 pounds to being 91. We project a 27 gram per day milk protein loss. Now the model is saying you're in excess. The model is saying I have excess 6, 7 pounds of milk, but in reality, your marginal return would be here. The same goes if you were going to take this diet and make it 85 pounds of MP milk or 84 pounds of MP milk. Model may be projecting a 6 pound loss. It may end up being 2 pounds. So a simple, I guess, rule of thumb, just given the data out there today, I would suggest when you're close to balancing a typical diet, if I change MP, I would take whatever the model predicts and multiply it times 20 to 25%, as you can see right here. So that would get you to 1.9 pounds. So I think I'll stop right here, see if there's any questions at this point, Benjamin.
Yeah, great. So I don't have any questions coming through here, but there's of course a slight lag. So since we haven't built any questions up to this point, I'll let you carry on.
Okay.
And I'll just encourage people, if you have questions that are coming up, let's put them in the chat. And the next time you take a break, then we'll carry on.
Okay. Thanks.
Thanks, Peter.
So the part 2 that I was going to talk about was looking at amino acids more specifically, not MP. So this is methionine marginal efficiency to milk protein. This was a recent publication by Dr. Mike Van Amberg. This is milk protein response yield, grams per day. This is percent of MP. If you do the calculations here and look at the effective change from 2% to say 2.5%, on the response here on the y-axis, you get 11% marginal efficiency from methionine to milk protein. So that means when you look down here at this barrel and stave, if I truly believe the barrel and stave, that efficiency should be close to 100%. I'm only getting 11%. So why, when I supplement methionine over this range here, is not more of it ending up in milk protein? Does this mean methionine is acting more as a stimulatory role? Is it acting more from a requirement role and we're just excess requirement? What is going on? And we'll come back to this here in a bit. But I think this is very intriguing to look at, in that you would think as I change a stave, it would be 100% or close to 100% after I track for catabolism during uptake. But no, it's actually 11%. So nutrition models, this is in CNCPS, they recommend looking at 1.1 grams per megacal of ME. And the efficiency of use there is 57%. The range in this data set was 1.7 to 5.4. So this is perceived as a requirement. What I would suggest is this is actually a response function. So when milk protein is lower in price, or rumen-protected methionine is a high cost to add to the diet, perhaps I want to be out here at 0.9 or 1.0. When milk prices are high, milk protein prices are high, perhaps I want to be at 1.2. This efficiency can vary. What's coded in the model is 57%. I'll just give an example here. So if I have a 90 pounds of milk, 95 pounds of milk, 70 megacal diet, and I want to be at 0.8, I would supplement 56 grams of metabolizable methionine. If I go from 0.8 to 1.1, I would add 21 grams per day of metabolizable methionine. If I use a synthetic source to do that, that would cost 54 cents to add 21 grams of metabolizable methionine. 11% marginal return to get into milk protein, I would end up with 0.23 pounds of milk protein, or 45 cents a cal a day. So in that scenario, I'd be at a negative return. So where you are on here, I think you need to look at what is the price of milk protein. If the price of milk protein is high, then yes, I may want to be closer to 1.1, 1.2. If the price of milk protein is low, and the price of feed protein's high, I may want to be at 0.9 or 1. Now, of course, there's differences in responses in fresh cows, peak lactation, and late lactation. This is just looking at it from a big picture. This is a simple study by Dr. Bill Weiss at Ohio State. It was done a number of years ago, and I think it's very intriguing to look at it from an amino acid standpoint. In this control here, so there was 3 treatments. This control was a corn protein-based diet where they only fed corn protein. Then they added methionine and lysine, rumen protected. And then this treatment 2 was your more typical diet with alfalfa, soybean meal in the diet. As you can see, same crude protein, same MP, more metabolizable methionine, of course, here, and then our lysine to methionine ratio would be more appropriate. So you can see, you're looking at this, I think all of us in using a model, we're going to say that this diet here in the middle should do the best, has the right ratio and has added metabolizable methionine. What the cows said is no. So the cows in treatment 2 with your typical protein ingredients you feed, there was an increase of 5 pounds of milk, 4 pounds of milk over rumen protected methionine and lysine. Energy corrected milk, 5-pound response here as well, 3 versus rumen protected methionine and lysine. this is what interests me here. So we have a 40-gram increase in milk protein yield. At the same time, we have less methionine in blood. So this is plasma methionine concentrations. You increase here with rumen-protected methionine. I don't have that in the diet, but I'm still getting more milk protein. So how are the cows giving more milk protein when methionine is perceived as being the most limiting amino acid? Methionine and lysine. And here's branched chain amino acids, and we're going to come back to this a bit. Again, they were much higher in this diet that had soy and alfalfa. So I think this is an interesting study to point out that just looking at methionine and lysine may not be telling the whole story. This was a field study. It was done in California on 20 commercial dairies. They went out to high cow pens. I think the high cow pens were averaging about 100 pounds of milk, and they took blood plasma samples across these 20 dairies. They took those blood plasma samples, analyzed for amino acids, then tried to find out if there's blood plasma amino acids that relate to production or better performance of higher milk protein, etc. Blood plasma has its issues with analyzing because it's not telling the whole story of uptake or blood flow, but it can be an indicator, particularly for methionine bioavailability, considered by some researchers. So what you can see here is methionine or lysine had no relationship to milk production. When I looked at the ratio, this ratio is all over the place. I have ratios from 2.5 to 4.4 in this data set, and again, no relationship to milk production. So does the ratio of amino acids arriving at the intestine or the ratio of amino acids in the blood delivering to the mammary gland, does it really matter or not? And do these limiting amino acids matter? Looking at this data set in the blood, you would say no. Other issue, when we talk about predicting milk protein yield, we've seen a number of studies or several studies like this that pointed to an issue in our models, and it relates to energy. So when I feed more energy, this is looking at a previous study, and I'll refresh you guys here. This is low energy, low protein. This is high energy, low protein, low energy, high protein, and high energy, high protein. And this is milk protein grams per day. As you can see here, when I feed more energy, I got increase in milk protein here, despite not changing the protein concentration in the diet. The models are not predicting that same increase. And you can see that here. In both cases, changing energy content increased milk protein. The models did not predict that change. You can see even when you get out here to these high protein, the models were predicting some very different results than what we observed. So what's going on, or what's new in amino acids beyond just looking at amino acids by themselves as requirements? Well, I would suggest nutrient signaling or control of protein translation is sort of a different way to look at it that research has been looking at for the last 10 to 15 years. And one example I like to give is we think about de novo milk fat. And de novo milk fat, for a number of years, we used to look at acetate or butyrate and think about, is acetate or fiber digestion limiting de novo milk fat? Is that why we see differences in de novo milk fat? Well, when we found CLA and realized that a small amount of certain CLAs arriving at the mammary gland can dramatically affect enzyme activity, fatty acid synthase, SOAP1, we realize there's substantial control of regulation of de novo fat synthesis. For milk protein, there does appear to be some control as well. It's highly integrated, which makes sense when you think about life. This molecule I'm talking about, this regulation of mTOR, it's been widely shown across a number of species. This is a citation you could look at for that. It's been shown in vitro with cells from a mammary gland. It's been shown in rat pups. And then this was a study that showed in dairy cows. And what's being suggested is that this factor here called mTOR integrates signals such as hormones, such as insulin, So if there's a change in insulin status, it can integrate a response and affect the rate of milk protein synthesis. Same for amino acids. Certain amino acids, particularly leucine, if the amount changes, it can lead to a change in activation of mTOR, which can lead to a change in milk protein synthesis. Same for energy. So if I have a change in ATP to ADP ratio, I can get a change here as well. So a crude way to think of it is you're speeding up the engine, or you're going from a V6 to a V8 engine. You have much more capacity to make milk protein. This is a better slide, I think, to suggest sort of what I was just talking about here, put together by my former advisor. Here's this point of regulation. I just talked about mTOR, amino acids, energy, and hormones. These are the ribosomes that make protein. Traditionally, we just thought about amino acids and what's limiting for the tRNAs. We didn't think about, can we change ribosomal activity? Can we change ribosomal abundance? Essentially, can we change the machinery that's used to make milk protein? And more research is saying that regulation exists in dairy cows. And I think we should consider this instead of just looking at supply, essentially the barrel and stave over here. This is a study that I did several years ago, and looking at that concept. So this is a control diet where we fed a lower metabolizable protein diet. This methionine, lysine, and histidine, we supplemented according to CNCPS and NRC recommendations. As you can see, that increased milk protein percent, much like we'd expect. We also supplemented isoleucine and leucine. You did not see a change in milk protein percent, and then we supplemented both together. What was interesting is that milk protein yield increased for both sets of amino acids independently. So supplementing isoleucine and leucine, we have this increase in milk protein yield, so milk protein pounds per day, independent of methionine, lysine, histidine. So how are we increasing milk protein yield when methionine and lysine should be first or co-limiting? And we just showed a response from methionine and lysine. Are we stimulating the machinery in the mammary gland, stimulating mTOR to higher anabolic state, higher milk production? We may, and that may be the reason for this response. And then together, we had an additive response in milk protein yield. Again, one comment I should make here, this is a very small study, small sample size, 8 cows, short-term study because of infusion. This does need to be repeated, but it was sort of our first, I guess, a study to look at this concept in more detail. Milk production, again, so when we supplement isoleucine and leucine, we see this response in milk yield, also energy-corrected milk, about 4 pounds, and then together we see the greatest response, about 6 pounds over control in terms of milk and energy-corrected milk yield. So why is this occurring? Not going to go into more details related to amino acid concentration in blood or uptake. I would encourage you to check out the paper, but the one thing I wanted to point out to everyone is methionine concentrations in the venous. So the venous blood is what exits the mammary gland. So arterial brings amino acids here, the venous blood exits. Here is the control. This is methionine, lysine, histidine, this isoleucine and leucine, and here's the combination. What you see is you can see a dramatic change from about 7 micromoles to close to 24, so much higher was leaving. So only a small portion of the extra methionine we supplemented actually got taken up by the mammary gland, net uptake. When you go through the calculation, it ended up being about 12% increased methionine uptake when we actually increased supplies much more than that. So the methionine uptake, only a small portion of it is being captured. Most of it is leaving in the venous. So does that support other functions? Does that get catabolized? Are we over the requirement? Are we under the requirement? I think it points to some more questions here. So the marginal methionine efficiency in this diet, we just look at MKH. This is marginal methionine to milk protein, was 9%, very close to what we see here on this data set. When I added in isoleucine and leucine, that increased to 20%. So it's a nearly double efficiency because we had an additive effect on milk protein. But again, a very low efficiency, much lower than would be suggested by the barrel and stave. A look at amino acid efficiency of use. This is the same set of treatments here. Methionine is going to be very high, 90–95% when it's not supplemented. So when methionine was deficient, most of the methionine being supplied was ending up in milk protein. When methionine was supplemented, that efficiency dropped to 60%, 65%, very close to where CNCPS has a fixed efficiency. You look at lysine, we see the same thing. When we added lysine, the efficiency dropped, and the same thing for leucine and isoleucine. So what I'm saying is here, we consider this to be fixed efficiency over here in the barrel and stave, when in fact there's clearly variable efficiency of metabolism. The animal can take up more methionine or take up less methionine depending on our needs in the udder. One other more recent study related to isoleucine and leucine. This is milk protein yield. This is milk pounds. They had a negative control and a positive control. Again, we can see this increase here as I'm adding different amino acids. Methionine, I have a response. When I add isoleucine and leucine, a milk protein yield, I have a tendency, and over here, I have an increase in milk yield. Now again, this lower producing cows here, I think this is an international study, but we do see this increase from isoleucine and leucine again. This meta-analysis recently published as well found leucine to be associated with increased milk volume and milk protein yields. Methionine was the only other amino acid to be found to be associated with in this meta-analysis. So I think there is some evidence out there pointing to leucine having a role. When we look at it from a requirement standpoint, we'd probably say no, but is it having a stimulatory effect and therefore having a role in production? So back up a little bit to the study I just, my PhD study I discussed, how do cows increase milk protein when methionine may be limiting? And one of our hypothesis is amino acid transport. This is a leucine transport model that we did in vitro, but we tried to mimic in vivo concentrations extracellular. What we found here in this study, 72% of the leucine that went into the cell exited the cell. So it was a very rapid bidirectional exchange. This bidirectional exchange is a cell using that to match up the right amino acid profile intracellular. So if a cell is short on leucine intracellular, there can be less efflux. If the cell less efflux, and there also can be more transporters pulled to the membrane to pull in more leucine. There's flexibility to match where you're at. If methionine is short, there can be less efflux, and you have less in the venous blood, like I showed in my study. If it's in excess, you're going to see more exit. This is an example of the intracellular pool. This is leucine turnover here. Just want to show how rapid that turnover is of the entire pool. So in 7 minutes, this entire pool of leucine was turning over intracellular. So I don't have the best terminology to say it in one sense, but amino acid transport, I believe, is a mechanism, and it's been suggested by previous researchers for a number of years to match your intracellular needs, say at the tRNA level, that precision, given imbalances that may arrive from diet and in the blood, it can adjust amino acid profile. We followed up the in vitro work I did. Dr. Xinbei Wang did an in vivo amino acid transport study, and she had found similar findings. So for leucine and isoleucine, 40% of it was being effluxed. So of the leucine that went into the cell, 40% exited. Methionine and lysine, 20%. So you had this influx, efflux, and this can vary matching what the cell needs. So I just wanted to touch on this amino acid transport a little bit because I don't think it's a way that the animal and the udder can modify amino acid profile arriving at the udder. It can adjust uptake, it can change efficiency to match the precise needs to make milk protein. So part 2 for conclusion, I think we need to think about not being like a machine or an engine. So in this case, an airplane, if I change the amount of inputs, something happens. I mean, the plane could crash. For a cow, if I change inputs, nutrient inputs, she has flexibility. Think about early lactation. There's tremendous flexibility to meet the needs. She can change efficiency, she can mobilize nutrients, etc. So when I start thinking about, say, lysine requirements or methionine requirements, think more of it as a response or variable efficiency. And there's a lot of, you could say our efficiency is low in our model, so there's a lot of flexibility there. And in case of methionine, and the model has a 60 or 55% efficiency. Well, that could range from 90% to 10% at the marginal when you change the amount of methionine being added. So think about not just think about as a response surface and the flexibility associated with that. And secondly, this nutrient signaling. Certain amino acids can stimulate milk protein. So that aspect of it, I think, should be considered. And then amino acid transport. So I think at some point we're so concerned about the profile, the proper ratio. Well, that ratio can get adjusted during absorption, during catabolism by the guts. And then when you get to the udder, you have amino acid transport, which can dramatically affect that profile as well. So part 2, application, when we're looking at formulating diets, it was from a recent proceedings paper by Dr. Hannigan, predicting milk protein yield. And this is the equation he was using. This may be what's upcoming in NRC, but I'm just basing this off publicly available information here. But these were the factors in the equation: digestible energy, which when you think about energy and you think about mTOR, so you have an energy that stimulates, and then you also have the energy that's needed to make milk protein. Body weight was in that equation, digestible NDF, and then 7 essential amino acids. So in this case, we're not just looking at methionine and lysine, we're looking at 7 different essential amino acids, and each of them could independently increase milk protein in this equation. So taking what this data set, and I think this is based on 950 treatment means that came together with this equation for predicting milk protein. When we're out formulating diets on a day-to-day basis, this is one of the first things I would look at or recommend looking at today. Fermentable carbohydrates or starch. I'm either going to get microbial protein, or I'm going to have more energy substrates being in metabolism, which then will stimulate milk protein. So increasing fermentable carbohydrates is something I would suggest. Multiple essential amino acids, maybe first looking at total essential amino acids, and then realizing that there's multiple essential amino acids that may drive milk protein. We look at synthetic amino acids, lysine as an example. Synthetic lysine shouldn't be just compared to the lysine value, say, in soybean meal or lysine value in blood meal. We have to compare all essential amino acids in that feed protein. Amino acid efficiency varies, nonlinear, considered marginal economics. So when I'm feeding that high level of methionine or feeding the methionine according to the model requirement, if I feed less, the efficiency may change, and it may still support adequate milk production. It could be less, but it may be much less than what you would consider. Consider protein regulation and formulation. So there's flexibility. So we know milk fat prices have been substantially less. So if I have less concern about decreasing milk fat, maybe I feed more starch or overfeed starch to drive this. Essential amino acids, hormones, from a formulation standpoint, can't really manipulate that, but we can change insulin downstream. So if I change the amount of oleic fatty acid, I may change insulin in circulation, which then may change milk protein. This last point, didn't talk about this much with amino acids, but I do think this is important, but not part of this talk. So I'll go, is there any questions at this point?
Yeah, Peter, we've got several questions that have built up here, and so we'll sort through them. The first one early on was just for some clarification. When you were showing the model outputs and talking about taking a predicted MP milk and multiplying times 20 or 25%, you meant the number of pounds in excess of, or the improvement that you made in the ration, correct?
Yes, that's correct.
Yeah,
And that's at a normal diet. If you have a diet that's formulated at 12% crude protein, then that 25% probably should be much higher. But a typical diet, say 15 to 17, 18% crude protein, 25%, 20% marginal efficiency, I think is a safer assumption than using the 67% in the model.
Thanks. And then I think along those lines, a question, curious if you had any comment, when we try and evaluate research on different bypass amino acids, how do we interpret peak or area under the curve of plasma concentration or actual milk produced, milk protein? Because it's difficult to integrate different research that we see on different products.
That's a very good question. I may defer some of my comments. I mean, you have experience in this area as well, Dr. Winter. But first of all, I think just looking at milk protein, it can be a signal. I don't think it should be used to calculate absolute bioavailability. So if I see a change in milk protein from feeding product X, I think we can infer that the product is working somewhat, but I don't think it can be used to calculate absolute bioavailability. Area under the curve, I think is an improvement from I guess, area under the curve. Are you talking about a pulse dose on a single day, or are you talking about, yeah, assume a pulse dose? In that case, I would say there are some major concerns when I pulse dose methionine and uptake by tissues that can change. Catabolism can change. So calculating amino acid bioavailability under that, it can be a tool, but the absolute is probably not.
Thanks. And then one other question for right now, at least, regarding when we try and think about the variable efficiency of MP, is there not yet enough data, maybe from a few different papers, LaPierre, Deppel, a couple others that you've cited, is there not enough to build a larger model for that variable efficiency? And maybe what's holding us back in that area, in your opinion?
Yeah, that's a good question. I think Dr. Luis Moraes at Ohio State, he's put forth a publication 2 years ago. And I think that model is somewhat along these lines, is what Doppel and others have looked at. As far as in-a-ration software like NDS or AMTS, I think we should look at it. Some of the European models, such as the Norfor system, do use a variable efficiency. So I think there's data out there to make a step in that direction. Of course, that's going to require our existing models to change, and they may be changing here, particularly with the next NRC. So I don't know if that answers your questions, but that's sort of, there are people already doing that, is a statement I would say, and I think it could be incorporated into our models, which then I think would enable us to look at optimization quite differently.
Yeah, thank you. I think that'll do for now, especially because we're a little crunched on time. So I'll cut you loose to go through the rest of your slides here, and then we'll see if there's any lingering questions.
So part 3, and this is more just outside the box, extreme. I mean, I can sit here and we can say milk protein is going to be the highest it's been since 2014. But this picture here on the right, I think there's a number of producers across the country experiencing this. This is 5 tankers of milk, dumping milk in the state of Florida. So if we're called upon to reduce our production by 15%, or we have no cash flow, what should we do? So I wanted to go through a couple studies looking at short and long-term effects of manipulating milk, protein concentrations and formulation. This study out of Penn State, and there's been a number of studies related to this topic, they fed a blood meal product, or they fed feather meal, and over at about 4 weeks to 5 weeks, they've seen a change in hemoglobin concentration in the blood. So hemoglobin is a marker for histidine status. So as you see here, the hemoglobin's higher when we're feeding the higher histidine. Without, this starts to decline. This will have some effects on intake and also milk production. In this case, there was a 6.6 pounds energy-corrected milk change over 10 weeks. Dr. Alex Ristoff has done a number of studies looking at this, and when you look at it in a Latin square, 3 to 4 weeks, there's usually no effect on intake or production from histidine. If I look over 10 weeks, we see changes, and that's what's shown here. So a general comment to make is, okay, so if I have a 3-week, 4-week period where I must reduce feed costs, or I must change production, perhaps I should consider reducing the amount of histidine that I feed in a diet for 3 to 4 weeks. If I go longer than 5 to 6 weeks, I think we should realize we're going to be depleting muscle tissue reserves of histidine. That will have detrimental effects on production, detrimental effects potentially on peak milk, as well as lactation curve possibly. So just something to think about, that you may be able to go a couple weeks and be okay on histidine by reducing the targeted histidine in diet formulation. This was a study done by Dr. Jeffrey Zanton, and I think his question was looking at in a short-term Latin square design, which this is the 4 weeks here, versus a continuous randomized block design of 16 weeks. When we feed low protein or we feed adequate protein, do we see differences in our results? In this study, they saw no difference. So when I fed diet for 4 weeks, or I fed for 16 weeks, 2 different levels of MP, I think it was around the adequate MP was around 17% crude protein. I think this is around 15. You do see a 7-pound difference, but what I want to point out is this difference between 4 weeks and 16 weeks is the same, and there's no diet by week interaction. Same on milk protein. So this study would suggest that, yes, if I reduce MP the preceding 4 weeks, I'm probably going to lose production. And these, again, were high producing dairy cows here, around 120 pounds of milk. Coming back to what the topic we just talked about, our marginal efficiency, I think is interesting to look at here is, if I was using CNCPS or NRC and I made this change from a low MP, from an adequate MP to a low MP. So I believe they went from 11.5% of dry matter to, I believe, 9.8% of dry matter. I would have predicted a 16-pound change in milk yield. What actually was observed was 6 pounds, and it would be very close if we use this publication of 20%, 5 pounds. So again, if I do really pull MP back, if I reduce MP by several hundred grams to save money, I'm going to lose milk, probably in the short term, but it'll be much less than what the model would suggest. This is a study done about 10 years ago, and they were looking at when you restrict feed intake, you restrict energy intake, protein intake. This is about 100 days in milk. Do you get the same change in metabolites, mobilization of body tissues to do early lactation? The reason I put this study in here is I wanted to look at how quickly the cows recover. So these cows, again, about 100 days of milk, they're restricted about 50% in intake. So they're only fed about 50% of their intake. You can see dry matter intake drops and it recovers. This 3-week period was the period of restriction. Milk yield, as you can see right here, you have this decrease, but then it recovers, and we have similar milk yield here. So again, you could recover back to where I was. So in an extreme example, I may be able to dramatically reduce, which will affect production, but the cows were able to recover. This was around 100 to 130 days of milk. Different stages of lactation, you may get a different result. Milk protein, same thing occurred. One other study I want to look at, and there's very few studies like this, is a long-term study done by Dr. Chris Reynolds. It's not published, so I was using what I could find online, but he fed 3 levels of protein. These are the changes in the diet, 215 cows. He fed for 3 lactations. The outcome, 16% crude protein diet was the best, but what I wanted to point out, there was no difference in days of pregnancy. So changing protein over a whole lactation had no effect on conception or days of pregnant. Feed costs, fed replacements were highest. So in the low crude protein diet, there were more cows leave at the end. There was more attrition. If you want to know more about this study, I would look it up here. What I found interesting about this study, looking at the data, is yes, milk yield and milk protein changed just like we would expect. When I look at milk protein yield, and this is a percent weeks, there was a 44-week lactation. 95% of the lactation, the first lactation cows had significantly less milk protein on the low crude protein. Compared to the third lactation at 39%. Milk yield, very different there. So if I'm going to feed a low protein diet, a very low protein diet post peak, I may want to target second or third lactation and not first lactation animals. I'd encourage you to check out the link. There's very nice graphs there showing this, but I think it's interesting is if I change protein, reduce, and first lactation may affect the whole lactation versus third lactation only affect 40% of the lactation. So one other, just to summarize early lactation, carryover effects, and apologize, I'm running out of time here, but we've had Alex Tebby talk about this today, but there's been several studies out there show significant responses in fresh cows. We also have this review paper here, most studies showing a carryover effect of 3 to 9 pounds of milk. So what I do in this fresh period may carry over into the rest of the lactation. So when I'm looking at fresh and peak lactation, there's probably a milk response from MP. Is it 4 pounds? Is it 8 pounds? It is 10 pounds. I think we could debate that. There's likely a positive ROI to continue to feed high MP, high amino acids to fresh cows. Post-peak, I think you need to evaluate ROI. So this income here in milk protein is going to change depending on milk protein prices and also feed protein, and that may change. Situation we're at today, we may not want to be supplementing as much MP amino acids post-peak. Close-up dry cows, I'm going to skip this for lack of time, but big picture, and I think this somewhat surprised me when I was putting this together, we have above average milk protein prices, at least compared to 2016 to 2018. If you look at the cost of metabolizable protein via sesame, it's below average to this time. So I have higher milk protein prices and lower input nutrient prices. So if I don't have a liquidity issue and I have no milk volume constraints on the farm, I probably want to formulate diets with equal or enhanced, probably enhanced concentrations compared to 2016 to 2018, and look at protein regulation. Can I feed more fermentable carbohydrates and starches cheap? Can I look at multiple essential amino acids instead of just methionine and lysine? If I'm in an emergency or significant financial constraint situation, can we target certain groups of cows? So fresh cows maintain our protein amino acid supplementation. Low cows, particularly mature cows, can we cut significantly back in protein and amino acids? And I would argue yes. Evaluate marginal responses. So I'm trying to predict the economic impact of changing MP by 200 grams. Look at some of the marginal efficiencies that I discussed. Again, consider this. Can we feed more starch to accomplish greater milk protein? And again, evaluate impacts short-term versus long-term. Just to summarize here briefly, consider marginal responses. Changing MP amino acid supplies may change performance. I don't believe there's a fixed requirement. There's a marginal response surface. We look at balancing for amino acids today, look at multiple essential amino acids. I would recommend tracking total essentials first, and then consider individual essential amino acids such as methionine, lysine, or histidine. We definitely want to do this because both nutrients are relatively cheap today, RDP and fermentable carbohydrates, and then consider the flexibility of metabolism. And lastly, think outside the box. So with that, I'll take questions. And sorry for running over a little bit.
Hey, Peter, you're fine. It was good to listen to you today. I got one question that's already in the box, and we've got time for maybe one or two before we're going to have to cut off and go to the social hour. Someone's curious about, from an NP standpoint, so we know we can cut it back and we may not lose milk, but what happens if we cut it back from a repro standpoint? And do you have any perception on what might happen to us on the repro? And, let's say we sacrifice NP for a month.
Yes, I don't have any answers to that specifically, but I would encourage whoever's asking that question to check out this study by Dr. Chris Reynolds over 3 lactations. They didn't see any differences in days to pregnancy, but they did, and I don't know if it's significant because I can't see the stats. There was a pretty large difference numerically in abortions. So I would check out this study if you can. There was another study review by Sinclair et al. In 2014 that looked at that. I don't remember the findings of that. So to answer your question, I think it should be looked at, and I would encourage you to look at this study right here.